Catalyst tonight, preparing for the next big outbreak.
Are bats carrying the next deadly virus?
Unlocking our genetic past.
Behind this door is where they unravel the mysteries of ancient DNA.
And the surfing scientist checks out some high-tech submersibles.
This is one of 3,000 robots monitoring the world's oceans.
G'day, welcome to Catalyst. It's well known how devastating Ebola can be. When it spreads from animals to humans, the consequences are very often fatal. Well, here in Australia, our fruit bats can harbour a similarly deadly disease called the Hendra virus. Now, it's mainly affected horses so far. In fact, in 15 years, just 3 people have died from it. So, for the moment at least, the chances of a human infection are fairly low. Although if you do catch the disease, there's only a 50% chance you'll survive it. So just how much do we know about the way that Hendra spreads, and are we prepared for a major outbreak of the disease? Marianne de Macy investigates.
This is the Australian Animal Health Laboratory in Geelong, Victoria. It's the largest biocontainment facility in the world.
This is where you remove your clothing, okay?
As in all of my clothing?
All your clothing, so you're staying there starkers.
Even my underwear?
Even your underwear.
Oh dear. Biosecurity doesn't get much tighter than this. Anything that goes in must be decontaminated before it comes out, including me. This facility houses live viruses, often unidentified, lethal and highly contagious. Viruses for which there's no treatment and no cure. If anything escapes, it could spell disaster.
We have to do strategic research so that we will be ready if any virus happen to get into Australia.
In here are the worst of the worst when it comes to viruses. Many names may be familiar to you, like SARS, Nipah and Hendra virus. And when they strike, they hit the headlines.
The rarity of this deadly virus, confirmed outbreak of Hendra virus.
Scientists are trying to stay one step ahead of the next outbreak of a deadly virus.
Viruses come from all different sort of reservoirs, but one thing is certain now is that most of the human new infections originated from animal.
This story began in '94, when a mysterious viral outbreak caused the death of a horse trainer and several of his horses in the Brisbane suburb of Hendra.
3 adjoining stable complexes near Brisbane's major racecourses are under quarantine after the death of 10 horses this week.
Teams of scientists worked around the clock to isolate and identify the new virus. They called it Hendra.
It took 2 weeks. Now, I think by any international standard, that's a world record. And you can just compare the discovery of the Hendra virus with SARS virus. I mean, SARS was circulating and killing more than 100 people before the virus was discovered 4 months later.
An electron microscope gave scientists their first picture of Hendra. It made the cover of the prestigious journal Science.
So these are examples here, are they?
Yeah, so this is the original Hendra virus, for example. Alex Hyde did a very high resolution shots there.
So that's Hendra virus, that's the real deal.
Armed with this information, the next challenge was to find out where the virus originated.
The starting point for the hunt for the origin of the virus, if you like, really was the paddock where the first horse got sick. So we were looking at possums, bandicoots, rodents, birds, a range of things that were in the paddock there, and found no evidence of infection there.
The death of a second person who handled an infected horse in Mackay raised the stakes.
Tests have confirmed that a Queensland man who died at the weekend had the same mystery horse virus suspected of killing trainer Vic Rail.
Finding the source of the virus became more urgent. They were closing in on a host. Clues pointed to an animal that could move swiftly and cover a lot of ground.
Within, I think, probably 6 weeks in central Queensland, we were able to identify antibodies to Hendra virus in bats.
This significant discovery didn't come as a surprise to Limpa Wang.
We're finding more and more virus from bats. Although the first bat virus was discovered 100 years ago, but I don't think people have paid enough attention to bats as a reservoir of nasty virus.
These viruses have always been there. The reason why we're now seeing them, but they're now spilling out, is not that the viruses have changed, but that the opportunity for the viruses to spill out has changed.
Zoologist Les Hall has been studying bats or flying foxes for over 40 years.
And they're like hanging out by the river?
Yes,
things?
they're the little red flying foxes there, and that's a typical roosting area for them. Flying foxes are becoming more urban. It looks as though it's urban areas are more reliable food resources. So that does increase the amount of contact with humans.
But the presence of more bats in urban areas raises more questions than it answers. Why hasn't there been an increase in people being infected by bats directly? And how do horses become infected?
Flying foxes excrete virus in fetal fluids, in urine, and also in saliva. We believe that those fluids from an infected flying fox contaminate grass or feed or a water trough or a stable rail. If the horse is susceptible to that infection, then it will become infected.
Researchers are now trying to prove that theory.
Now, it's just before dusk and the researchers are out there laying down these plastic sheets which they'll put under the bats to collect urine samples overnight. Now, potentially these urine samples may contain live viruses, so that's why they're all kitted up in those outfits. Now, tomorrow morning they'll be back at first light to collect these samples and take them back to the lab.
They process the urine samples, searching for any signs of Hendra virus. Suspect samples are sent to Lympha's lab in Geelong for further analysis. Their challenge is to find out how it gets from bats to horses.
It's basically a lottery, 1 out of a million chances of a bat jumping to your horse, and your horse is susceptible. Laboratory conditions, we have done that experiment once and we failed. So it's very difficult to understand that process.
Hendra seemed to disappear, but more than a decade later, it struck a third time last year.
When we arrived here in the morning, we noticed that, the staff noticed that there was something very wrong with the horse. He was quite dangerous and very maniacal. He was just uncontrollable. And, it was obvious he was going to die.
This is the yard where the first horse that contracted Hendra lived. Subsequently, another 4 horses fell sick with the virus. At least 15 staff had high exposure to the horses, but only 2 became infected.
I would have to say probably myself was the one that was exposed to more of the horses than anyone else, and I didn't become infected. Now, I don't know whether or not the horse actually sneezes on you. If you're unlucky to be in front of the horse and it sneezes at that point in time and you get inhalation of the virus, we don't know.
Tragically, Hendra's third victim was David's employee and friend, Ben Canine. Once a horse becomes infected, Hendra multiplies exponentially and becomes far more virulent. And while few people acquire the infection, when they do, it's lethal, with a 50% mortality rate.
We can't do anything right now. We don't have drugs, we don't have vaccine. We are confident that if there's a will, we can develop a vaccine which can be utilized either in horse or a human. And we have some preliminary data actually in animal models. We already have a candidate vaccine working. The challenge is there's a lack of commercial drive.
So the science is there, but the money's not.
That's right.
It's a job made even more difficult by the fact that there are very few labs in the world like this one equipped to handle live viruses.
You need to take a 3-minute shower, then I'll meet you outside.
It's time for me to leave the lab. Everyone is required to decontaminate to avoid viruses escaping the facility.
So, despite all the research that's going on in there, there are still a lot of unanswered questions.
So if bats are the natural reservoir for so many deadly viruses, should we eradicate them from urban areas?
No, I don't think we need to be afraid of bats. Diseases emerge because of an upset of the balance of nature, an ecological impact. And Hendra has emerged, at least in part, because of that impact on the environment of bats. Killing more bats is simply going to upset that balance even further and arguably make the problem worse.
So do we face the threat of a new deadly virus from bats?
Without any doubt, I think there's a chance that you have the virus which is even more deadly and more readily transmissible between humans. Now, if these ones jump out of bats, then we will get much more serious outbreaks.
We're about to gain a much deeper understanding of our past and possibly even our future, thanks to a new study of some very old DNA. A world-leading research unit at the University of Adelaide has taken genetic material from bones, plants and soils. They've looked at a variety of samples from Tassie tigers to HMAS Sydney's unknown sailor to Homo floresiensis, better known as the Hobbit. Paul Willis unlocks the doors to our past.
More creatures are extinct today than are alive. What they were like as living animals will always be a bit of a mystery.
If you want to find out what life was really like for fascinating beasts like Diprotodon here, or how other creatures evolved and became extinct, perhaps you want to find out about human remains from the more recent past.
Then no longer do we have to rely solely on the fossil record.
Ancient DNA is critical. It's very difficult to do, but it's critical. We've got to do it.
We can tackle questions from 10 years ago, 100 years ago, 10,000 years ago, and 100,000 years ago.
But extracting and analysing ancient DNA is a painstakingly complex task. To explain why it's so difficult, let's go back a step.
DNA is the blueprint of life. It's like a barcode that contains most of the information we need to identify who we are. Hair colour, body shape, personality, each of us has our own unique barcode, and each species has its own unique DNA.
When we die, DNA slowly degrades, as there are no longer any repair enzymes doing their job to keep us going. It becomes scrambled, lying in a crumpled heap, and most ceases to exist.
By the time this process is finished, there's really not a lot of DNA left to play with.
That is until the Australian Centre for Ancient DNA at the University of Adelaide was set up in 2006. It's the only facility in the Southern Hemisphere studying ancient DNA, and it's considered to be the best in the world.
We had very little or nothing effectively in Australia, and so the potential was, let's bring in ancient DNA and start looking across the Southern Hemisphere for evolutionary information.
This is the Ancient DNA Lab, and they're so concerned about contamination, I'm not allowed to go in. Not that I'm taking that personally.
We left it to the experts.
We work kind of like in a virus movie. You know, you see everyone in bodysuits moving around, but we're doing the opposite. Rather than trying to stop the material infecting us with some horrible virus, we're trying to stop our DNA getting onto the specimen. It's basically an exercise in uncontrolled paranoia. And the reason for this is there's very little DNA left. I mean, minute traces of damaged, tiny, broken pieces of the original genetic material. And that's because over time, water, oxygen, temperature, bacteria and fungus are breaking down the DNA.
It seems they have a somewhat impossible task to extract and analyse DNA.
The bone sample, we have to grind it up to create a powder to actually pull the calcium out of the bone. Then that allows us to extract DNA from the remaining bone powder. We may only get 10 copies of that particular gene out of an entire gram of bone.
Once they've got their ancient DNA samples, they bring them here to their modern DNA lab. And I'm actually allowed into this one.
Obviously, we can't work with just 10 or 100 copies of a particular piece of DNA. There's just not enough of it there to do anything. So we need to use a process to actually copy a particular gene.
It's a bit like taking a fragment of barcode and making lots of copies of it so that it's easy to work with.
We've gone from having 10 copies of this gene to 1 billion copies of this gene, all in this one little tiny tube.
Once you have your fragment of DNA, the next step is to compare it on a database of the gene sequences of a number of known organisms. See if you can get a match. And that can actually be done on the internet.
In a bat of a virtual eyelid, the team can quickly figure out whether the DNA they have is from an ancient creature or from modern contamination. The centre covers a broad spectrum of research, from living Tasmanian devils,
how Tasmanian devils lost genetic diversity, and that's really important for trying to understand the impacts and the origins of this devil facial tumour disease,
to ancient mammoths. So this would be from the Northern Hemisphere?
Yep, it's from Canada, from the Yukon, and you can see here we've actually been sampling for DNA. There's actually about 3 or 4 experiments worth in there, carbon dating as well. That's the chunk that we've cut out of it.
But it's not only creatures of the past. Jeremy has also been instrumental in trying to solve the mystery of the identity of HMAS Sydney's unknown sailor. The Sydney sunk off the coast of Western Australia in World War II after an encounter with a German raider. The sailor's remains were found in an unmarked grave on Christmas Island. From the few fragments of uniform that could be found, the Navy could narrow down the identity to 3 sailors. But because the body had been so exposed to water and the passage of time, DNA analysis was difficult.
We had to apply our high-tech sort of ancient DNA techniques. We managed to extract DNA. Unfortunately for those 3 shortlisted men, we didn't get a match with the maternal relatives.
But the hunt goes on. Identifying people from 70 years ago or 18,000 years ago is all in a day's work for these scientists. And like Jeremy, Alan is determined to break the code. He's been working with an international group on the mystery of the Flores hominids, affectionately known as the Hobbit. Is it a mutated human or a new species?
We want to get the genetic answer. So our challenge was to try and get traces of DNA out of these very valuable specimens. The bone material of the hobbits was quite degraded. The teeth are the things that tend to survive the longest, and so we'll often get DNA from inside the tooth. So we sampled the interior of teeth from 2 of the most complete hobbit specimens.
But because the sampling had to be done in Indonesia, contamination was a big problem, and the DNA analysis was unsuccessful.
The 2 specimens we looked at were 18,000 years old and about 15,000 years old. The hobbits survived right up until 11. So I'm very hopeful in expeditions which are going on at the moment, they're going to find a young specimen. The younger it is, the more chance we have of DNA survival, as a general rule.
Analysing ancient DNA is a very tricky business, but it may help to complete the puzzles of the past.
It's the only way to understand how we've ended up where we are now and what the likely future holds.
In 1999, Australia launched 10 submersible monitoring devices off the coast of Western Australia. They were part of an international effort to keep a watchful eye on our oceans. Well, today, there are literally thousands of these Argo floats patrolling the seas, and they're finding surprising new results about how our oceans are responding to climate change. Surfing scientist Ruben Meerman reports on the real-time data streaming in.
We know the ocean has a huge impact on climate. Sea surface temperatures affect cloud formation and rainfall. But until recently, the processes that go on deep beneath the surface have been largely a mystery. But that mystery is being slowly revealed. 10 years ago, Australia released the first Argo floats into the Indian Ocean. These nautical robots dive up to 2 kilometres every 10 days and then surface to transmit their precious measurements to Argo headquarters. 20 countries maintain the global armada of more than 3,000 Argo floats, and they're providing oceanographers with an unprecedented wealth of data from deep beneath the sea.
Well, this is an Argo float. As you can see, it's in pieces, but you'll get to see what it's made of. So this is the sensor package, which measures temperature and pressure and salinity. And here's the antenna that transmits all that good information from the surface to the satellite system. And that all sits within this guy, the pressure case, and that has to be very strong because this float undergoes very strong pressure changes, like it gets up to 200 atmospheres down there.
So Susan, how does the float work?
Well, when the float needs to sink, all it does is run this pump here and it pulls in oil from the outside bladder, which reduces its volume and it'll just fall down through the water. When it's time to come up and profile, it'll pump again and pump some oil out into the bladder, expand the bladder and just float on up to the surface.
Simple Archimedes principle.
Exactly.
Argo floats have completely revolutionised oceanography because now we can peel back even the remotest parts of the ocean and take a look underneath.
So this is showing where the floats are. That's the yellow dots. And those are the tracks of the float. So that's all.
When they analysed that flood of Argo data, Susan's team found the ocean's upper layers are warming 50% faster than previously thought.
The thing about warming in the ocean is that it controls partly sea level rise, because as the water warms, it expands. And so the sea level rises where that heat is built up. And so now we have a much more accurate picture of what's contributed to sea level rise over the last 60 years, and I think more confidence about future projections.
According to current CSIRO data, sea level has increased by 5 centimetres over the past 15 years and is increasing at a rate of more than 3 millimetres per year. Using Argo's salinity probe, scientists can also deduce rainfall over the oceans, where ordinary rain gauges can't go.
What we expect as the planet warms is that the rate of evaporation and rainfall is going to get stronger, so the hydrological cycle is getting stronger. And so what we're seeing is as we use Argo salinity data, the places where it's already wet are getting wetter, and the places that are already dry are getting a lot drier. And Argo is going to allow us to track that into the future.
The Antarctic Circumpolar Current is the strongest current system in the world and is known as the engine room of the world's oceans and climate.
We used Argo measurements of temperature and salinity and compared them to observations from ships dating back to the 1960s. And what we found is that the Southern Ocean is changing. It's warming more rapidly than the rest of the global ocean.
Another robotic device, known as a glider, is supplying oceanographers like Dr. David Griffin with valuable information on currents.
The beauty of the glider is that it fills in the gap that Argo is in the deep ocean but doesn't measure the continental shelf. That's where we really need observations of what's happening in the ocean. So the glider can profile the top 200 metres many times and measure the really powerful energetic currents that are happening over the continental shelf. At the moment we have 3,000 Argo floats all around the world. Imagine if we had 3,000 of these controllable floats all doing what we want rather than just sampling where they happen to be.
Increasing storm activity, warming waters and rising sea levels. Now, at least with the Argo system, we have accurate real-time data of the changes taking place.
Next week on Catalyst, Paul Willis uncovers ancient life in Chile.
These are the world's oldest mummies.
And take a peek through one of the world's most powerful microscopes.
This electron microscope can see things smaller than an atom.
Thanks for watching Catalyst. I'm Graham Phillips. See you next week.